Our next presentation is Affyimmune Therapeutics, and I'd like to welcome Daniel Jantz, CEO, to the stage.
Great. Good afternoon, everybody, and thank you for your attention. My name is Daniel Jantz. I'm the CEO of Affyimmune Therapeutics. Affyimmune is a clinical stage autologous CAR T-cell company, and we're addressing the clinical unmet needs in solid tumors. Our first indication that we're going after is in advanced thyroid cancers, and our second indication with the same product will be in non-small cell lung cancer. So today I'm going to cover 5 topics. So first I'll introduce you to our lead asset, AIC-100, which is a CAR T-cell directed at the tumor-associated antigen ICAM-1. I'll then present a high-level overview of our first indication in advanced thyroid cancers and really highlight where the unmet need is as we see it within that particular indication. I'll cover some clinical data, the early clinical data that we've seen in phase 1, and highlight why we at Affyimmune, as well as our clinical trial site heads and PIs, think why we have a bona fide drug in hand. Lastly, I will talk through and highlight our team and why we think today we have the right team put together in order to advance our therapeutic into further trials in phase 2 and beyond. So Affyimmune is targeting the tumor-associated antigen ICAM-1. ICAM-1 is an intercellular adhesion molecule, and it's expressed at a low level across multiple cell types, including endothelial cells, epithelial cells, as well as immune cells. Our CAR T-cell construct, AIC-100, uses the natural binder of ICAM-1 as the CAR construct. It is an integrin known as lymphocyte function-associated factor, and integrated into the CAR, it confers a number of biological benefits to our CAR T-cell. First and foremost, LFA1 is actually expressed on T-cells naturally, and it's the interaction of LFA1 on the T-cell with ICAM-1 on endothelial cells, which actually facilitates the transmigration of natural T-cells across the endothelial layer and into the tumor stroma. And so we take advantage of this natural biology by integrating LFA1 into our CAR, which then facilitates the enhanced translocation of our CAR T-cell across the endothelial layer and into the tumor stroma. We've also optimized LFA1. LFA1 on our CAR, we've optimized it so that it has a low on-target off-tumor toxicity. And so the affinity and the binding of LFA1 on ICAM-1 does not, as we've tested in our preclinical models, show that it has any cell killing on those cells which have a low expression of ICAM-1, including endothelial cells. And this is translated, and this is seen when we look at our clinical data where we have an exemplary safety profile. In addition, when we tested our CAR T cells in our preclinical models, we don't see any tonic signaling. And so with that, we don't see any associated T cell exhaustion. Lastly, as part of the natural biology, when our CAR T cells migrate to the tumor and engage ICAM-1 on the tumor, you get the natural expression of or excretion of cytokines, including interferon gamma. Interferon gamma upregulates ICAM-1 on the target tumors in the tumor microenvironment. So this creates a feed-forward effect in terms of upregulating the target for our CAR T-cell. And then it also addresses a central challenge for a lot of antigen-targeted therapies, namely the challenge of interpatient and intratumoral antigen heterogeneity, which we're able to address with our system. In order to track our CAR T cells, we have integrated the somatostatin receptor into our CAR, and this allows us then, with the administration of gallium dotatate, to be able to see and visualize where our CAR T cells track to within the tumor, as well as see their residence across time with the duration of the therapy. So I want to touch a little bit on the unmet need that we see within thyroid cancer. So broadly speaking, there are multiple histologic types of thyroid cancer. And over here within the differentiated thyroid cancers, we've grouped a number of patient subpopulations, including papillary, follicular, oncocytic, as well as the partially differentiated thyroid cancers. For most patients, through thyroidectomy and radioactive iodine treatment, the patients do well on that therapy, but those are not the patients that we focus on at Affyimmune. We're focusing on where there's the highest unmet medical need, which is the 15% of patients which are refractory to radioactive iodine and have also progressed on multiple lines of multi-kinase inhibitors. We're also focusing on those patients which have anaplastic thyroid cancer. So this is the most aggressive form of thyroid cancer, which you see on the right. And these patients don't really have any therapies available to them for treatment. And so within these two patient populations, these patients have about 90% of these patients express ICAM1. And so from a target patient population perspective, we're able to capture about 6,500 patients per year in this group. Our initial positioning for the anaplastic population is in first line for those patients which are BRAF wild type, and then in third line for those patients who are relapsed and refractory to the multiple lines of multi-kinase inhibitors. So moving on to the clinical data, these are the data which excited us as we were seeing them, and I think really positions us as potentially a best-in-class solution within the field. So what I'm showing here are the data which we made public earlier this year. It's 6 patients worth of data at our dose level 2 and our dose level 3. I don't show our dose level 1 data because that was subtherapeutic, and there's a few things that that are takeaways over here. So one is we see overall a disease control rate or disease control in 4 out of 6 patients, and we see overall responses in 2 out of 6 patients. This includes the partial response that we see on the right in the first ATC patient, and importantly, in the patient on the far right, by RECIST criteria, we see a partial response. But in this patient as well, what we observed through FDG PET scanning was a metabolic complete response, which from our understanding was a first that was seen for a CAR T cell, an autologous single-dose CAR T cell for solid tumors. And so that was something that was broadly exciting for us in terms of thinking about how do we translate this into our phase 2 trials. Response is a proxy for survival, and so it's really that's where we've been focusing our time in terms of understanding and generating more data. So from a survival perspective, these patients, by the time they come to us for the treatment in phase 1, these are incredibly sick patients. And so this is where, from a survival perspective, these data are really exceeding our expectations. Again, as an example, in the anaplastic population, which you see on the right, the median overall survival of these patients when they come to us is anywhere from 3 to 5 months from a median overall survival perspective. So the fact that we're seeing patients which are surviving, as you can see on the bottom right, for 22 plus months, patients that are still alive today. Second patient, more than 10 months, also still alive today, is something that's exceptional. In the advanced differentiated thyroid cancer populations, we've seen patients who are alive now for 18 and 19 months. We've also learned from the patients that we were unfortunately unable to treat. So on the left, you see 2 patients which unfortunately progressed and passed away. And the learnings that we have over there is that we really need to be able to treat the patients as quickly as possible between their enrollment and infusion. So unfortunately, with these 2 patients, they both had delayed infusions, one because of personal reasons and one was just structural. And with these delayed infusions, unfortunately, they passed and were unable to survive. And so we're really taking away with these lessons with with the lessons learned from these patients, putting them into our phase 2 trial design. So one is, fortunately, we're able to remove the stagger that we have in phase 1 from a safety standpoint. We're also removing the need to test for ICAM1 as a marker because of, with these patients, 90% of these patients have ICAM1 expression. And then lastly, we're also apheresing conducting pre-enrollment apheresis. So again, shaving off days between when patients are eligible for treatment and when they get their infusion. And so those are all things that we're looking forward to. Just to make the charts a little bit more tangible, I'd like to show just a few patient scans. So on the top right, this is the scan showing our ability to track our CAR T cells. And as you can see over here, we're able to track it over time. And so this is with the gallium dotatate staining. We're able to see residence of AIC100 at the site of the tumor, both at baseline on day 0, as well as 6 weeks after that time. And so with this, we're able to understand and see the persistence of our product within the patient. On the left, the first set of slides, that's the FDG PET scans that confirmed that complete metabolic response in our patients. And this was at our dose level 3, or 500 million cells. And on the right, or to its right, that's by RECIST criteria, we had the partial response. And this was at our dose level 2, so only at 100 million cells at RECIST criteria. So with these data and these responses, coupled with the safety data that we're seeing, where we've not seen any grade 3 or higher adverse events, whether it's CRS or neurotox, we really think we have a drug in hand or a product in hand that may support a best-in-class profile. I just wanted to end with sharing with you the team that's made this all possible. So Sonal Gupta has really spearheaded all the clinical work that we've done within Affyimmune, including our thyroid and non-small cell lung cancer programs. Pete Jelinas has come to us. He heads up our CMC, and he really reinforced the importance to us on prioritizing every aspect of CMC, highlighting for us that for a lot of cell therapy companies that have unfortunately not progressed or failed, for many of these companies, it was because of some of the challenges they experienced on the CMC side, so we continue to prioritize our CMC work. Don Buchanan has led our work on the ClinOps side, including managing clinical trial sites across MD Anderson, Northwestern, as well as City of Hope. We made a decision early in the course of our company, a strategic decision to bring in quality quality leadership into the organization early. And so Arya has that broad experience across large pharma as well as small companies, and he has oversight across both our clinical as well as our manufacturing functions to really drive home and take leadership on making sure that from a product perspective, we're adhering to all the quality standards within the industry. And we have Josh over here who heads up both our finance and operations. So with that, I wanted to end the presentation. If there's anything if our story interests you and excites you, we'd be happy to share more about our story either after this talk. And we also, I didn't have time to touch on our non-small cell lung cancer program, so please do feel free to reach out. We're happy to tell you more about our story.